Good evening, and welcome to the Leukemia and Lymphoma Society of Canada's first webcast called Lymphoma in Research, Yesterday, Today, and Tomorrow. This presentation is part of a series featuring the latest advances in blood cancer research. Let's start the presentation. My name is Andy Fertino, Regional Director for the Quebec Division. I will be the host of this online event. In the webcast, you will hear only my voice and our speaker's voice. Since there are many of you online, questions may only be asked at the end of the presentation using the chat function online. The presentation will last 45 to 60 minutes and will be followed by a 15 to 30-minute question period. The presentation is also being recorded. You will be able to listen to it again as it will be available on our website. We will send you an email with the link once the presentation will be online. Our mission at the Leukemia and Lymphoma Society of Canada is to cure blood cancers and improve the quality of life for patients and their families. We offer support to people affected by blood cancer every step of the way. Please reach out to us if need be. We have several informative tools about lymphoma on our website. The Leukemia and Lymphoma Society of Canada currently finances 41 research projects and invested a total of $36 million in research since 1955. More than 138,000 Canadians live with or in remission of a blood cancer. Lymphoma is the most common type of blood cancer in adults. It affects more than 43,000 Canadians. At this time, I'd like to introduce our speaker, Dr. Jean-Sébastien Delisle, who has generously accepted to spend part of his evenings with us. Dr. Delisle is an hematologist at the Maisonneuve-Rosemont Hospital Research Center. Since October 2010, he has been a clinician scientist at the hospital's bone marrow transplant group and has led the research program in cancer, immunology, and transplants. He is also an associate professor at Université of Montreal. Over to you, Dr. Delisle.
Thank you very much for the kind introduction. Start by saying that we won't be able to cover everything in an hour, but I will do my best to make sure that all the major categories of innovations in transplantation, in lymphoma research and treatment are covered. As Andy said, I'm a bone marrow transplant physician and researcher. And I'm involved very much in research and clinical development, including development of novel therapeutics in the field. So we need to go back to biology if we need to understand how lymphoma forms and how we can treat it. The blood system is a system built like a pyramid that is like lying on its side on this slide. So everything starts with a stem cell that gives rise to myeloid precursors or lymphoid precursors. The myeloid precursors will eventually form the platelets, the red cells, and most of your white cells. The lymphoid precursor will give rise to the lymphocytes. And lymphoma is a cancer of the lymphocytes. So you see the series of arrows there. It indicates that several stages must be completed for a precursor to become a mature cell. And the cancer process can occur at several times along the way. Introduction to cancer biology, and probably as the only thing most of us need to know, cancer revolves around four fundamental concepts. Cancer is a cell gone wrong. Problems can arise in four biological processes. The self-renewal process, whereby a cancerous can propagate by keeping its ability to generate progeny. We call that self-renewal, and this is the entire concept behind the idea of a cancer or a leukemia or lymphoma stem cell. Then, a cancer is a cell that has a problem with differentiation, that cannot fully differentiate to mediate its normal function. So block in differentiation is a salient feature of several cancers. Related proliferation is probably what is most known about cancer, which is often pictured as a disorganized growth. And apoptosis is cell death. So cancer cells not only need to proliferate to survive as cancer cells and cause all the damage they cause, but they also have to learn to avoid cell death. So all of cancer biology can be summarized by these four concepts here. So if we apply that to lymphoma development, you can see here on the far left the common lymphoid precursor that will progressively differentiate into B cell. That's the top row. The B cells are the cells that secrete antibodies normally. And on the lower row in red, the T cells are remove or killing infected cells in the body, sometimes even cancer cells in the body, and have a very important role in immunity. We call them T cells because they mature in the thymus, which is a gland that is situated in your thorax. The B cells are called B because they perform their initial maturation steps in the bone marrow, B. So, to become highly specialized immune cells, the B cell precursors must do something that is extremely dangerous. They have to break their DNA, rearrange it, so that their specific receptors, the B cell receptor or T cell receptor, function. Each time you play around with your DNA, there is a risk of error. Errors in genetic code are at the very core of any cancer. So there is a moment during the lifetime of a B or a T cell where these cells are extremely vulnerable to cancer transformation. This is very important. These receptors, B cell receptor, and this is an example of the B cell receptor, it survives, proliferate, and mediate its normal actions. A cell needs to receive signals from the external world through its BCR. The thing is true for the T cells. And cancers of the lymphoid system, the lymphomas, learn how to use these signals to their own advantage. So, in no activation of this very complex cascade of events that follows the triggering of the B cell receptor, that becomes dysregulated in cancer. And we will see later how these become, fortunately for us, treatment opportunities. So, BTK is a molecule that will become very important for the growth of certain lymphoma that is now targeted with a drug called Brutonit. The PI3 kinase complex is targeted by a drug called idelalisib. And the mTOR complex can be blocked by a drug called everolimus. There is now a way to target the B cell receptor pathway in lymphoma, and we will see what are the advantages of that and the potential limitations later on in the presentation. So, does lymphoma develop? The honest answer to that is we don't know. It would be fair to say that most lymphomas arise because bad luck occurred while they were playing around with their DNA. And that chance, meaning the likelihood of not forming a good DNA rearrangement, is probably at the core of most lymphomas. However, certain deficiencies in the immune system that are inborn, that are genetic, may predispose to lymphoma. Acquired immunity deficiencies such as HIV infection or immunosuppression following transplantation will predispose to lymphoma. Some autoimmune diseases, lupus, will also increase the risk of developing lymphoma during the course of the disease. Some infections, viruses such as hepatitis C or Epstein-Barr virus infection, or HTLV, human T-cell lymphotropic virus 1, lead to lymphoma development. Chronic inflammation in the stomach through a bacteria called Helicobacter pylori, which is responsible for most ulcers in our world, can predispose also to the development of certain types of lymphoma. Obviously, the big unknown category is made of chemicals and drugs. We know that radiation and chemotherapy, sometimes used for other cancers, predispose to lymphoma. We know that certain drugs used in rheumatoid arthritis, for instance, such as methotrexate, which is a chemotherapy agent, or tumor necrosis factor inhibitors, can also predispose to lymphoma. The big unknown here is the contribution of pesticides and other toxins in our environment, and these have an important role in lymphoma development. My suspicion is that we will learn more and more in the next few years about the impact of our environment on cancer development in general and perhaps lymphoma development in particular. The lymphoma is a cancer of the lymphocyte. The lymphocyte isn't restricted to one organ. The lymphocyte's job is to patrol the body in search of infections. Lymphoma can arise anywhere. It can arise in lymph glands, and there are lymph glands all around the body. It can arise in the spleen, which is like a big lymph node in the abdomen. But it can also arise in areas that are not lymphoid tissues, like the skin, for instance. And it has the potential to migrate everywhere. So even if a lymphoma presents at various places in the body and presents as a vastly disseminated disease, it doesn't mean that it will be harder to treat. Sometimes yes, for certain lymphomas, but not in all cases. It's not like a colon or breast cancer that has a very, very, where the distribution has a very important impact on treatment outcome. So, this slide is on purpose very complicated. It is to show how good we have become at making categories for lymphomas. So B-cell lymphomas are 85% of all lymphomas, and those include Hodgkin's disease and most post-transplant lymphomas, while T-cell or NK cell lymphoma are only 15% of the number of lymphoma cases. And you can see from this very long list, many entities are very rare. As a practicing hematologist, I can bet that I will not see a case of all these entities. Some are so rare that it is first of all not seen very frequently and not studied very much, which can be a problem for the patients who suffer from these conditions. However, we are starting to know more and more about their biology, both sides, B and or T cell lymphomas, which guides the development of new therapeutics. So, lymphoma treatment is highly connected to biology. We have to realize that all lymphomas are not created equal. There are varying response rates, and biology will determine the proper course of action. Biology will determine what kind of treatment we can envisage for the future. It is very important to study the biology of these diseases in order to design and validate improve and more precise therapies. So, an important concept, and I alluded to it before, is that the type of lymphoma depends on the stage of lymphocyte maturation. So it is connected to the normal biology in some way. And the maturation, and this is the example of B-cell lymphomas, the maturation is related to the anatomy of the lymph node. Here that the lymph node has a marginal zone, a mental zone, and a germinal center zone. So many of you might have heard the term mental cell lymphoma. It means that the cell that has become a cancer is from the mental zone. A mental zone lymphoma is the same thing. Now, we need also to consider that the biology of diseases, of lymphomas that look like, be very different. In red is B-cell CLL, chronic lymphocytic leukemia. The most common leukemia in our societies. We can see that if the cancer cell is a stage of development that precedes the maturation in the lymph node, we call it unmutated variable gene. Well, if the cell is from a cell that has completed its second stage of education in the lymph node, we call it CLL also. I can show you, and I will show you next slide, that these two diseases are not the same at all. Same thing with diffuse large cell lymphoma. Diffuse large B-cell lymphoma is the most prevalent of all lymphomas. And that is circled in green. The types. One is AEC, activated B-cell type. The other one is GC type, germinal center type. So these two diseases look exactly the same under the microscope, but they have a different biology. So how was that discovered? If you look on the right panel first, the case of diffuse large cell lymphoma, the interesting modern art type graphic on the top is what we call a heat map. It represents gene expression. Green or red, depending whether the gene is upregulated or downregulated. And by looking at, or well, in this case, gene expression, so RNA, of several cases of lymphoma, it became clear, if you look at the patterns of green and red, that despite the fact they were looking the same under the microscope, the diseases were very different according to the genes they would express. And you see on the lower panel the impact it had on therapy. The germinal center B cell do much better than the activated B cell. So for diseases that look like the same thing under the microscope, there are vast difference in the biology, and that's reflected in treatment. So that's why we need these markers to make sure that we can specifically address the problem in this case of activated B-cell lymphoma to make sure that the response rates in the future will match germinal center B-cell lymphoma and that both curves will actually move up. Now, if I bring you to the left side of the slide, you see the difference unmutated versus mutated cell as on treatment. That's different. Here is that we need to be careful because these methods to classify disease and the impact on treatment are always based on the treatments available at the time. So it's possible that some of these markers or drugs have no impact. So this is also why developing new therapeutics is essential. If we stay on the topic of chronic lymphocytic leukemia, perhaps for another minute, there is a gene anomaly, 17p, the loss of 17p, which carries a very important tumor suppressor gene called p53. It is known that 53 deficient disease is highly resistant to chemotherapy. It is much less resistant to targeted therapy or immunotherapy. It's treatment, knowing that your patient has a disease with a given marker. Just to reinforce the point, how important it is to characterize the disease from a biological standpoint, to do and design anything intelligent about them, about treatment. This is another example. This was a paper published by my colleague, Nathalie Johnson, who practices at the Jewish General Hospital in Montreal, who did that while she was in Vancouver a few years back. You see here the impact of the expression of two genes in diffuse large B-cell lymphoma. MYC gene, high proliferation. It's like the gas pedal. Well, CL2 is that prevents cell death. There are two key concepts in cancer biology. If at the same time push on the gas pedal and you have the brakes through IBCL2 expression, you get a red line very, very poorly. These are unfortunately highly resistant to chemotherapy. So, for patients, we need to devise or we need to approach them differently because we know that standard chemotherapy will not do the trick. So this is an active area of research, but it highlights once more the importance of performing precision research in the biology of lymphoma. So, the treatment principles in lymphoma haven't changed for quite some time. We need to appreciate here the complexity. There's a wide variety of diseases and a wide variety of treatments. In general, we need to divide the world of lymphoma into two entities, the aggressive versus the indolent or slow-growing lymphoma. Speaking, although this is not always true, aggressive lymphoma can progress rapidly but can be cured with standard approaches like therapy or radiation. Indolent lymphoma are slow-growing. We don't need to treat them because it will grow so slowly. However, if you give them chemotherapy, you may get responses, but almost never a cure. So we need to decide, does the disease require treatment or does the patient want treatment? And treatment is, since the 1950s, pretty much chemotherapy. And I'm going to tell you in the next, in this little while, how this is pretty changing. So, Chemotherapy is not a static field. Therapies we use now to treat cancer are very different from what was used in the 50s and 60s. And always discovering new drugs. There is a funny example. Bendamustine was discovered and used by East German chemists and physicians, and that was completely unknown to the Western countries. Until the Berlin Wall fell, it was rediscovered recently and has been a phenomenal drug to treat many lymphomas. We always need to keep in mind that chemotherapy does not only belong to the ancient world. It is very actual. It sure has a great future. All chemotherapy, which work at very high doses, in fact, is now used to treat refractory lymphomas. And I will tell you how giving more chemotherapy can be effective if you can circumvent the main complications. There are obviously targeted therapies that are taking the world by storm, and immunotherapy, my own personal favorite field, which involves pharmacological agents, biological agents, and cellular products. So we will cover that in the remainder of this webcast. So, first of all, we need to make sure that we approach this and our expectations about this are well measured. There are from time to time innovations that change the way we treat the disease, that are so radically different from what we have done before that it changes our treatment algorithms. I will tell you how perhaps the use of genetically modified immune cells is one of those groundbreaking innovations. However, if we look at recent history, it's something to realize that most lives have been saved through small increments, small and steady improvements in the way we administer chemotherapy. So it does not sound very, very sexy, but forming high-quality clinical research base. The best example of that is pediatric leukemia, which had 4% survival rate in 1950, 80% survival rate in our times. Non-breaking here, carefully designed and conducted clinical trials, agents of chemotherapy, altering the doses, changing a bit the regime. This made a lot of difference. Innovation that came up in the last 30 years now, 3 years, is the use of chemotherapy to treat refractory lymphomas. The problem with chemotherapy is that when you give too much, you kill the endogenous bone marrow. Extension, you kill the patient. So one way to circumvent that limitation is to collect the cells of the patient before administering high-dose chemotherapy and put in the freezer, basically. Obviously, not any kind of freezer, you will realize, and you need to add a product so that the cells do not form water crystals and get killed. But once you have the cells in the freezer, you give this very high dose of chemotherapy then administer the stem cells who have not seen this very high dose chemotherapy. These cells are technically still healthy and can rebuild the immune system that was the blood and immune system that was destroyed by chemotherapy. So, this is a way, let's say, to bypass what would be a limitation to giving more chemotherapy. And that works. I mean, this is the famous curve from the Parma study published in 1995, where in aggressive lymphoma relapse patients, giving more chemotherapy alone to very poor survival, but giving very high dose chemotherapy with salvage transplantation increased to 50% long-term survival. We are performing this procedure routinely. First wave of immunotherapy. Now I will move in immunotherapy and keep targeted therapies for the end. The first wave of immunotherapy was the monoclonal antibodies. First IT, for sure. These are curves from diffuse large cell lymphoma. 4 years. The care for aggressive cell lymphoma was CHOP. CHOP is 4 chemotherapy drugs. Everything was tried in the 80s to make this better. And in 3, a paper said, you know what, all your original regimen don't work, CHOP is still the best. Until this came, rituximab, which specifically targets one antigenic determinant, one flag or one tag on lymphoma cells, which is called CD20. And that really increased the survival rates in diffuse large B-cell lymphoma. How does that work? So this will require an effort. I'm responsible for the drawing, I'm sorry, but this is a cell. The triangular form on top, which is CD20, exactly. So the antibody will bind to this, and then 3 things can happen. It perhaps triggers directly through internal signaling pathways. It can also use complements, which are circulating proteins that can regroup on the membrane of a cell and make a hole and kill the cell. Mechanism, though, for the effects of anti-CD20 therapy. So an immune cell will recognize an antibody-coded cell, and then in the context of infection is a bad sign. It means that the cell is infected. So by analogy here, it macrophages and NK cells, which are 2 cells of the immune system. Oh, there's a problem with this cell, you must kill it. So it does. So the antibody recruits the immune system in a way. NK cells stand for natural killer. Looks like the name of a biker gang, but that's really what they do. These cells are programmed to kill any cell that looks suspicious or seems infected. Now, we have expanded the CD20 paradigm. Now we have antibodies targeting CD30. We have antibodies targeting CD22. But we've done something more original with these antibodies. We created, this was originally done by several groups in the United States, something called chimeric antigen receptors, or CARs. These receptors do not exist in nature. They are purely synthetic. In fact, what they are a fusion between an antibody that will recognize the CD20 or the CD19, which are also expressed, which are both expressed by lymphoma, However, these will be expressed by a cell of the immune system. So it will tell the T cell to kill the cell that the antibody portion recognizes. This is very clever because T cells do not recognize infected or cancer cells this way. A way to redirect them and make them highly toxic to cancer cells. Analogous to that are the bispecific engagers. So you see here CD20 and CD3. CD19 binds on the surface of the cell, and through another portion of this molecule, it binds T cell, which is a cell of the immune system, and sends a signal. Found something, kill it. Triggers the T cell to kill the cell that was bound to this bispecific engager. The most common molecule is blinatumomab, which is mostly used in B-cell leukemia. So this is now this is a sketch from the New England Journal of Medicine describing what CAR T cells are. So on the right side, you see a CAR T cell, so an external portion that will target a cell surface determinant, and a intracellular tail that will tell the T cell, oh, I found something bad, kill it. You can see on the left side that that's not how a normal T cell recognizes a target. T cells have a specialized T cell receptor, and it's a very, very fine and extremely precise mechanism to kill virus-infected cells or cancer cells. However, in some diseases like the B-cell lymphomas, we can take advantage of the fact that there is an abundance of targets like CD19 or CD20, and redirect the T cells toward that abundant target. So it is a way to use a very effective of the immune system even more effectively. So this is going to change some of our world. In refractory patients, it can bring complete remission rates at around in around 90% of patients. In B-cell lymphoma, the response rate is a bit less, according to the preliminary data that we have. It is not, although immunotherapy sounds very sexy and non-toxic, there are several side effects, and very severe side effects associated with the use of CAR-modified T cells. So there has been deaths secondary to the administration of these cells, and the other limitation is that it remains fairly complicated to make. So what you need to do is to take the cells from the patient, play with them in the laboratory, and you can see on the right side how complicated this can be. It is becoming simpler and simpler, although the manufacturers are getting better, but still it is complicated. So what's done basically is that you infect the T cells with a virus that resembles HIV. Obviously, it doesn't cause all the problems HIV does, but it's the same backbone, and it inserts the CAR gene into the genome or into the DNA of the T cell, so that the T cell will express the CAR at its surface and in a way be ready to kill. There is also an old immunotherapy where I devote most of my time when I do clinic. It is allogenic bone marrow transplantation. The immunotherapy is based on the fact that when you use a donor that's compatible but not identical to the patient, the T cells, the immune cells, from the donor will recognize the foreign cells of the recipient, kill them in a standard rejection reaction. This is very good when the targeted cell is a cancer cell, and we call that graft versus tumor effect. Risk and the problem is that it can also affect the normal cells of the recipient's body, and that's called graft versus host disease, which is associated with several, with a lot of morbidity and sometimes mortality, unfortunately. So this immunotherapy is the oldest, running since the late 1970s, and still is the most widely used immunotherapy to treat blood cancer patients. So, trying to be more targeted, Epstein-Barr virus is a virus that can cause lymphoma. 9% of adults are latently affected, meaning that we carry the virus without knowing it, and the virus is not causing any problem. But sometimes we lose control over this. We receive immunosuppression following transplant, for instance, or sometimes the virus manages to elude the immune system and transform into a cancer the cell that carries it. So that results in lymphoma. So EBV is the virus that causes mononucleosis, described as the kissing disease, as this depicted here. It stays in several cells of your body, but notably the B cells, which is the natural reservoir for this virus. So when something goes wrong, it is most often, but not exclusively, the B cell that can become a cancer caused by the virus. So this is a picture of what we are doing at Maisonneuve-Rosemont. So we are training the patients or the donor, in the case of bone marrow transplant, T cells in a specialized laboratory, a laboratory that is so clean that you could perform, usual manufacturing grade pharmaceutical synthesis in. So we train the lymphocytes, recognize the virus, and we re-inject them to the patient. So this is a case of a patient. You have to picture that the patient is towards the back of your computer and that the feet are coming towards your face. There's a slice through the thorax. You see in the middle the heart, the black material are the lungs, and you see on your left a part of the liver. The arrows indicate tumors that were resistant to chemotherapy and that were essentially gone a few months after we injected the cells from the donor that were trained, specifically recognized the tumor. So in this case, there is no graft versus host disease because the cells are trained to recognize the virus and only the virus. Are there other ways to stimulate the immune system? Absolutely. Now, there is a natural immune response against the cancer. Unfortunately, in the end, it fails. Is there a way to revive it? And what we have realized is that these T cells that are infiltrating the tumors are on the brakes, pretty much. The brake is called PD-1, program 1. It sends a signal into the T cells that says, you don't activate yourself, and don't kill the cell you're recognizing as cancerous or infected. Now there are ways to block the PD-1 receptor and in fact stimulate the immune system. This already changed the world in melanoma treatment, in lung cancer, and other tumors will follow as well. In our world of blood cancers, we have been extremely successful in Hodgkin's lymphoma, where such treatment can bring a 90% response rate in patients who have been refractory to anything else. However, this is still under study, and we obviously need we need to study more patients and more diseases before we can apply that to every case. So I will finish by talking about targeted therapy. So this is a slide I bring from earlier in the presentation, where I showed you that in the case of B-cell malignancies, you can target the dysregulated inside the cells that tell the cell to grow and evade cell death. So ibrutinib, idelalisib, everolimus, and now venetoclax, which can inhibit BCL2. And BCL2, remember, BCL2 is a protein that prevents cell death. So if you block that, you can trigger cell death in cancer cells. Other drugs, such as lenalidomide, which has been used in myeloma a lot, is known to change the environment of the cancer so that it facilitates the activation of the immune system. Now, this is very nice, and it offers options beyond chemotherapy. And these can be effective when chemotherapy fails. They are not toxic. They do carry some side effects. Some are very significant. And the thing is that these therapies are relatively new. We are learning that mechanisms of resistance develop, and we do not know very well how to use them still. More clinical studies are required to make sure we use the right combinations, that we use them at the right time during the disease process. There's a lot to be learned, but globally, we can say that we have more and more weapons to fight lymphoma. So ibrutinib, which has been, still is, in lymphoma treatment. This is CLL, chronic lymphocytic leukemia treatment, standard treatment, chlorambucil versus ibrutinib. I think the survival curves speak for themselves. These are progression patients who did not progress while under the drug, and you can see that ibrutinib does very, very well. So, cancer cells are very smart, and they learn how to evade the therapies. You can, let's take 100 cancer cells. If your drug kills 99%, that's very good. But on the chance that the one cell that has survived has developed mechanisms that prevent this particular treatment to work again. So there are several ways to avoid that, and it's probably to target several proteins at the same time. It's like we did with HIV. You know, using one anti-HIV drug didn't work very well until we combined 3 types of drugs together to reduce viral load. The same analogy can be used here. We will probably need to target several mechanisms at the same time with different drugs to get the best effects. So what I have to do, and this is my conclusion slide before I take the questions, I think that we need to acknowledge that the last decade or last 2 decades have been extremely positive overall. Survival rates have improved, and this is due, if not exclusively, to the discovery and implementation of new treatments. We don't know how to prevent lymphoma. If you implement a good screening program for breast cancer or colon cancer, you will catch the majority while they're not dangerous. That cannot be applied to lymphoma. So most of our progress is really through improved therapeutics. focus on the types of lymphoma that are hard to cure. We will need to do clever clinical studies to combine treatments in a rational and effective way. We need to make sure we limit side effects, perhaps through precision medicine, meaning that medicine that is based on the biology of the disease and where only the defective mechanism of the cancer cells are targeted. Obviously, nothing of this can be achieved without more research, both in the lab and in the clinic. So thank you very much, and I will now take your questions.
Thank you, Dr. Delisle, for showing us how research and treatments are truly advancing and how survival rates are improving. The Leukemia Lymphoma Society will continue to do its part to speed up discoveries of better treatment options. As Dr. Delisle just mentioned, we now open it up to questions. If you have any questions online, you can now type them in. We will be reading them out loud, and Dr. Delisle will be answering them. I will read the first question online here. My husband has AITL, which started as EBV in 2016. It's 6 months and 2 biopsies to determine if it was this horrible lymphoma. He went through 6 months of EPOC, which completed in February. Had his stem cell in March 2017, and then showed signs of lymphoma in early April. They went ahead with an autologous transplant in May 2017. He also has, you'll have to help me with this word, Dr. Delisle.
No, but I mean, I can summarize here. This is the course, and you understand that it is extremely hard to comment on a particular case. I can tell right away though that this is very typical of AITL. AITL is a T-cell lymphoma, angioimmunoblastic T-cell lymphoma, which is exactly that. It is very difficult to diagnose. It is fairly tricky to treat, and it is associated with all sorts of immune side effects. So here I see that in the case that there is a question about diagnosis again, and it is tricky because the disease triggers an immune response that is extremely aggressive sometimes, and sometimes it can be confused with an infection, and sometimes it is the lymphoma itself. So here the complete description here. It's a complicated disease for sure, and there are, and obviously I cannot make recommendations here or precise recommendations. What I can say here is that it reflects a bit what I said in the earlier on, is that it is extremely sometimes difficult to make a right diagnosis, and some entities are relatively rare and have probably not benefited from as much research as other diseases. Now, specifically, I can see here that there are probably other options for this disease, and that these need to be discussed with the doctor. But it does highlight that at one point, sometimes in lymphoma treatment, we need to be not creative, but we need to make and to balance all the options, and this is sometimes a difficult thing to do, especially in the absence of precise guidelines. Because I said the world of lymphoma is so vast that not all entities have been investigated enough. So again, we need to say that more research in many of these areas are required.
So the next question we have here, are we making any progress on screening or prevention for blood cancers?
It's an interesting question. I would say that the answer is yes and no. Some blood cancers can be caught early. Myelodysplastic syndromes are one of them, and that can be caught by a regular blood test. But aside from that, it's very, very, very difficult to catch lymphoma in its early phase. Usually it presents, lymphoma or leukemia, it presents as a systemic disease. So early diagnosis is a problem. Now, in terms of prevention, there are some susceptibilities or vulnerabilities that are genetically encoded. So it does, for some diseases, run in the family. Some of these are genetic. In part, we rarely see patients with genetic forms like we see in breast or colon cancer. So it is difficult to see it coming even in these patients. Now, prevention. Since we don't know what causes it, it's very hard to prevent. And there's an emerging branch in oncology research that focuses on the environment. I think that will be extremely revealing because there are hints in the literature that some exposure to pesticides may be linked to the genetic anomalies that will trigger the lymphoma genesis, or the progression of a normal cell to a cancerous cell.
We have another question here. Expect that more treatments will be available for blood cancers within 2 years. In your opinion, which ones will be most promising? Yes, I think more treatments will be available. There are new molecules coming in today, and some centers specialize in testing these new molecules. What also will be occurring in the next few years is us knowing how to use the drugs we already have, because some of the drugs are relatively newcomers. If we, like, just as an example, mention the Brutonit, these drugs are relatively new, and it is my opinion that we still don't know where they really belong in the treatment algorithm. So once we know where and when to use them, we'll achieve much better results. Now, my own personal bias is that the immunotherapies will kick in significantly in the next 2 years, and that even fancy therapies like the CAR T cells will be more available under study in specialized centers, but will be more available to Canadians within the next few years. Very much. We have great questions here, so we'll move right along. So the next question we have is, what are the symptoms when your white cells are low? When they're marginally low, not much. Most patients who present as a consequence of a low white cell count, which can be secondary to a myelodysplastic syndrome or a leukemia, present with an infection. White cells are the soldiers of our body. So when they're not in sufficient numbers, people will show up with infections. They show with infections or like mouth ulcers. They can show with skin wounds that easily infect. They can show up sometimes with serious
fungal infection or in the lungs. We've seen that. So I would say that infection is the most recurrent or to treat infections are the most prevalent sign. Vast majority, though, of our patients who present in the clinic with low cell count have none of these symptoms, however, and we just see it on the blood test.
Okay, another interesting question here. Is it important for people with rare lymphomas to advocate with research institutions and funding bodies for attentions to these disease?
Okay, I'll read this one for you.
You. Thank you.
So the abbreviation is ENKTCL, nasal type. So this is a lymphoma of NK or T cells that is found in the midline, often in the at the level of the face, so nasal type. It is a Epstein-Barr virus-driven lymphoma that is extremely aggressive. And my answer is a resounding yes. It's very important to study these rare diseases, and perhaps more importantly is to make sure that if we have a lot of samples to study or a lot of patients to study, we can make the relevant connections perhaps to other lymphomas and see what's in common. And from regrouping some rarer entities together in order to get a critical mass of tissue and samples to perform the type of biology research that is so informative on the biology of the disease. So it's just like for rare diseases or rare congenital diseases, I think that there needs to be some kind of advocacy, and what are the best ways to do that? I think that patients and their families, through their doctors and institutions, are a start. And your question will probably be heard either at the Leukemia Lymphoma Society as well, where the goal is obviously to leave no patients alone.
Thank you. This is another good question that gets frequently as well. Can you explain why research is taking so long?
Oh, yes. For doing research myself, in a lab, obviously, at the very start, 2/3 or 70% of all the ideas we have are wrong. So we need to test all our promising or so-called promising ideas and realize that most of them are wrong. Then, when you have an idea that is good, that is probably leading to something, you need to validate it in another model. A researcher probably needs to validate it too. And then you need to interest other stakeholders who can carry this forward, and then liaise with clinicians who are willing to eventually experiment this into people and patients. Access to research for patients is something obvious. And that's another place where advocacy should be put in place. Patients should have access and the right to investigational treatments. And that we cannot do alone. We try to implement as many research protocols in our clinical research unit. There are logistical, financial, administrative reasons that are so that we cannot run all the studies all the time for everyone. So these are, I mean, from the bench, from the very idea of the research question, to clinical implementation, there, as you can see, many steps. Research is taking some time, but I mean, on the bright side, I have the feeling that if you look over the course of history or recent history, the very good and promising ideas move faster than before.
Another great question we have here: do you currently have CAR-T trials recruiting patients?
Stay tuned. I cannot answer that with all the required details. The short answer would be currently, today, no, but the field is moving fast.
Next question we have here, is there an age limit to immunotherapy? What age and why? Same question for targeted therapy.
Those are all great questions. What defines what is the age limit is often the expected or measured side effects. After a certain age, no matter how healthy you are, the biological reserves are lower. So for bone marrow transplant, for instance, we know, although we are getting better and better, know that we cannot treat patients above a certain age. But that target is moving. I mean, before, no one was transplanted after the age of 50, and then it was 60, and now 70, and so this is a moving target as well. So for immunotherapy, I would not see an age limit, except perhaps for the most experimental ones involving, for instance, cells that are likely to cause very severe side effects. Obviously, the first goal of all this is not to hurt anyone with bad side effects. So the age bracket for trials is set according to mostly the expected toxicities. There are other variables, but this is it. For some of the immunotherapies, though, that are much milder, I wouldn't see an age limit.
Along the same lines, how do we determine which patient is the right candidate for immunotherapy? Does the patient need to have undergone chemo first?
Because immunotherapy is an investigational field, by ethical standards, you can only offer it once you show that the patient failed standard therapy. And standard therapy in blood cancers is immunotherapy most of the time. Now, some of the immunotherapies are so successful in other diseases, like, so successful relative to standard treatments, that they are moving into what we call first line. So the immunotherapy is the first thing you give to the patient. But for blood cancers, we are not there yet. Standard treatments must be given first. There's a question here regarding the type of relationship a patient or caregiver can have with the medical team regarding access to new treatments or research. I think that, and it's very personal, and not all medical doctors would agree with me, but I always appreciate having a chat with someone who brings a patient who brings an article or a new idea or or question the foundations of our approach. This may vary, and I think that overall, our field in hematology, especially doctors who have been treating blood cancers, globally, we have been more receptive to innovations and to investigations than other fields of medicine. Now, we need to know, and everybody needs to know, that good ideas on paper not translate into benefits in reality. I send you back to my analogy of the researcher having 75% of his or good ideas in the garbage, at least. The same is true for any potential good idea a patient or a physician may have on the next course of treatment. These questions need to be addressed into a scientific protocol that is monitored, that fulfills all ethical standards, and that obviously is analyzed through the most rigorous scientific methods. So this is, I think you should always raise the question of new treatments with your oncologist, knowing that these questions should only be addressed in clinical trials, big, small, but these attempts must be done under surveillance.
So another question we have here, is lymphoma or blood cancers associated with lifestyle choices such as diet or exercise?
We don't know. We don't know. I wish I could answer that. As a piece of wisdom, I would say that exercising regularly and eating all your vegetables is probably a good idea. That said, no strong correlation with bad habits as cancers. We just don't know yet.
Next question we have here, is there an organization that guides and prioritizes overall strategies for blood cancer research in Canada?
No. I think that there are several organizations that will guide the strategies. At the early phases of research, it is still very investigator-driven. So in our system, we trust the people doing research. We believe that these people will have the right ideas. Now, after an idea is showing promise, what are the next steps? I need to bring up the fact that ideas that are likely to make someone rich move faster than others. This is perhaps the dark side of our system, but a drug that someone can sell will move the ladder of clinical investigation faster than another approach. This is true, unfortunately, and what we needed is perhaps other resources and organizations that will take the promising ideas in research that are perhaps not associated with a high commercial value, but a clinical value, and support them.
Next question here, there's not that much time left. There's just a few questions left, so we'll try to go through them if we can have enough time. It's about collaboration. So the question is, how are researchers' learnings shared between all of blood cancer researchers in Canada?
Oh, that's very interesting. The world's changed since the introduction of internet. If someone has a good idea somewhere and publishes it, it will pop in my inbox instantly. So not only are we connected among Canadians, but we're connected to the world. In Canada, we have, and it's emerging, with networks where researchers can talk together, exchange ideas, and collaborate. This is a culture change. We are seeing this more and more. There's an emphasis on collaborative research, and this is already bearing fruits. Now, not only in the discovery of new drugs, but perhaps in creative ways to implement programs will make accessible new drugs in our particular system.
So we have time for one last question here, and I guess I'll go with this one over here. So where is the best center in Canada to be treated for T-cell lymphomas? With a very unbiased.
No, but I'm afraid I cannot answer this. Wherever patients with rarer lymphomas or T-cell lymphomas are treated, you can pretty much make sure that the physician treating you can easily connect with a center where expertise can be shared. I think that would be fair to say. So exchanging expertise and ideas is perhaps more important than the center per se.
So there's one last question here, and I think for the sake of time, we do have time to take this last question. All your questions will be answered. Our last question is, can you comment or give any information on APL, so acute promyelocytic leukemia, or any new treatments available?
So acute promyelocytic leukemia has been the poster child of targeted therapy. Since the 90s, we have done wonders with this leukemia that was highly aggressive, able to, a leukemia that can kill someone in hours. We have been better, and so much better that when the leukemia is cut in time, we have now survival rates in excess of 90%. So these treatments are evolving, and we are at the stage where we are now performing these clinical studies to modify a bit the regime so that it's a bit more efficient, less toxic, but overall, APL has been a great success story. But there's one little limitation. It is a disease that evolves super fast, so it needs to be cut right away, or sometimes it is impossible to provide good care. So treatment must be started very, very rapidly. But aside from that, when it is the case, patients do very well.
That was our last question. So I would like to thank all for your questions and your participation. I would like to also remind you that earlier today you received an email with an evaluation form. Please take the time to fill it out, and it will help us to better understand your needs for our future webcasts. We also sent you links to our website. If you want to find out more about our services or research programs, please feel free to go onto our website and check it regularly so we would update it with new dates for upcoming webcasts. I'd like to thank you once again for joining us. Thank you, Dr. Delisle, for your time, and I wish you all a very nice evening.